Start the slideshow now. Just let me know if people can see the slides. Is that good?
Yes.
Okay.
Right.
All right. Well, welcome to Zoomlandia. I guess I'm the last speaker. I want to talk to you today about genomically guided integrated translational research. So as some of you on the phone know, Radha, Kelly, maybe a few others, my lab has been focusing on translational research for the last 10 or 12 years. And all translational research means is taking something from the bench to the bedside, something developed in the lab, a tool, you name it, and putting it to use in the clinic or public health settings. And that comes with it the need to really be sensitive to the handoff of, say, an idea from the lab to someone else. And those sorts of transitions can be difficult. But as you'll see, I think with the right kind of focus and paradigm, these sorts of transitions can be eased a bit. So here is just an outline of what I'll talk about. First, talk about workflows for personalized genomic medicine, focusing on the use of biomarkers, trying to understand the causes of disease at the highest possible resolution. I know you've had lectures from others, so this may be a bit of a recap, but genomics can get us to that highest possible resolution space, as you probably know. Then I'll talk about how one might be able to characterize the genomically induced pathology that, say, an individual with a disease might have, and how one might be able to choose potential therapies based on the insights of that genomic perturbation. And if one is lucky enough to design a therapy, an obvious next question is, how can we test it out and see if it actually works, say, on the individual for which we have identified genetic cause for their disease. And this bears on this paradigm of N of 1 trials, and I'll talk a little bit about that. And many people have had success using N of 1 trials in the framework I'm describing. For example, Rady Children's Hospital right down the street has a program to identify genomic perturbations responsible for babies in the NICU that might shed light on pathologies that can be treated in various ways and have had success, as have many other people at UCSD, Joe Gleason, Kelly, Radha, others. And then I'll close with a few comments on how one might be able to aggregate data of the type I will argue needs or will inevitably be collected in studies looking at this translational workflow, and how one might be able to leverage some machine learning techniques to exploit them further. Okay, as you know, a couple of 3 papers will be presented on Thursday surrounding these themes. I won't go through those in detail. All right, so as we discussed, in developing a technology that will, say, be used in the clinic, that technology has to go through various stages of development. So, obviously the first thing one needs to do is come up with some kind of concept, then maybe test it out in humans just to make sure it's safe. This could be a drug, it could be a new device, whatever it is. And if it's shown to be safe, you then pass it on to the next stage, where you ask the question, is it efficacious? Is it eliciting the response that it was designed to elicit? After that, you might think more broadly about implementing the tool or the product clinically. This might require a different set of studies and a different kind of orientation. And then after it's been deployed, you just kind of want to keep track of how it's being used to make sure it's not, say, being abused or not doing things that weren't anticipated. And this can benefit the population at large. Now, if you have a single individual, say with a diagnosis that's difficult to make, say someone with an idiopathic condition, and we'll discuss those later, then you might have to go through this process but ultimately for the individual focus. So you'd have to make some kind of diagnosis. If in the context of trying to prevent someone's disease, you'd need to assess someone's risk before you could tell them how they could avoid that disease. So there are parallels with kind of personalized treatments or preventions to the developmental stages that a technology might have to go through at a broader level. So in the context of more personalized treatments or prevention, after you have made a diagnosis or assessed someone's risk, you'd have to come up with some way of understanding the diagnosis to identify an intervention or a risk reduction strategy in the case of personalized prevention. And then of course, follow through in stages that are analogous to what one might do in developing a technology generally. And should one be able to work through these workflows, then obvious patient benefit might arise. Now here is just a workflow for trying to go from start to finish, identify a patient with some issue, subject them to DNA sequencing to find a pathogenic variant, work your way through this process that I've described to uncover what the molecular physiologic consequences of that mutation are, to the point where you might be able to identify some kind of therapy and then ultimately test that therapy out. And if you had success treating that patient, make the information available so that other patients might benefit. Now, in today's climate, unfortunately, there's many sources to identify patients. There's no shortage of patients, sadly. So that is not a big problem. DNA sequencing and the identification of pathogenic variants is possible, again, through sequencing. Identifying the molecular physiologic consequences of a sequence variant, not trivial to do, but techniques are emerging that we'll talk about. Identifying the best possible therapies or a possible therapy, again, not trivial, but people are having success. I'll describe a few of those successes. And one of the papers that'll be discussed Thursday describes major success in pursuing this for a particular individual. Testing out the drug to see if it actually does what it was anticipated to do is possible, and obviously it's certainly possible to put data back into the public domain. So with respect to the availability of resources and constructs to carry through on this, again, patient identification sadly is no problem. I think sequencing is getting easier, though as I'll point out, and as you know, it can be complicated in various contexts. Building databases, not a huge issue, again, with today's resources. Carrying out studies to prove that a drug works on a single individual, I'd say we're getting better, but still there's some uncertainty there with respect to the best way to go about it. Finding a drug based on a molecular perturbation of some sort or another, again, we're getting better, but we're far from having perfect solutions. I think where most of the action is just understanding the molecular or gross physiologic consequences of, say, a pathogenic variant to the point where we can intervene. Just point out that, it is possible to pursue many of these things in parallel instead of in sequence, though I've basically described things here as though this is truly a sequential process, and for the most part it is. But the goal of these studies, and we talked about these in a paper a little while ago, a colleague of mine, is to not only settle scientific questions, but ultimately benefit the patient, and that's the translational focus. Much of this research is of academic interest, but ultimately, if you want to put it together, the goal is to benefit patients. Okay, one of the papers we'll talk about on Thursday describes a success story going from start to finish. I won't deprive the presenter of going through the details. I'll just point out the various steps they went through paralleled what I described in the previous slides. So they sequenced the individual with the rare condition that they were interested in and ultimately found what they thought was the causal or pathogenic variant in the individual. They understood, and you'll hear more about this on Thursday, what the consequences of that pathogenic variant were to the point where they came up with the strategy for treating the patient. Essentially, there was a problem in the genome where suppressing certain activities of genes was important, so they made an antisense oligonucleotide to go in and bind to the region that was causing the problems. They built that construct and then tested it out in the patient by tracking various markers in the patient to see if it worked, and had success. So again, you'll hear more about this on Thursday. And I'm going to talk about these different stages in the remainder of my talk. Now, before I do that, there is something very interesting going on in the community, the biomedical science community, that's relevant to this workflow. And I'll just tell you about it briefly, call attention to it. And that is that the community is shifting from standard ways of creating and deploying medicines, which go under the heading, these weird headings, officinal. So the officinal way of making medicines is to create them in advance, put them in little bottles, and then distribute them to pharmacies, all around the planet. So if someone is diagnosed with a condition, they can go to the pharmacy and the pharmacist can, go to their shelves and provide the medicine to the patient. So this approach is more or less about stockpiling medicines that are made in advance of any patients actually coming in with the diseases. So they anticipate the use of these diseases. Magistral production of drugs, on the other hand, has to do with creating the drugs in real time. So you don't stockpile them in advance. You have a patient, you figure out what's going on with them to the degree you can do this in real time, and this doesn't work for everyone, so I need to emphasize that. And then once you come up with insight into what it is that, say, the patient is suffering from that could be treated, then you cook up the drugs right at the time. So think of making the drugs more or less at the bedside instead of going back and reaching into a stockpile of drugs. Now, why is this important? It's important because many individuals, say, won't respond to drugs that are made to treat everyone. So we're in an era of personalized medicine. And if it's the case that medicines need to be tailored to the unique features that individuals have, then you can't stockpile everything. You'd have to stockpile drugs for everyone on the planet. That's not going to happen. So instead, you have to figure out what's, say, causing a patient's illness, and then on that basis, make the drug in real time. And there's many examples of kind of magistral production of drugs, as given in this table taken from this paper in Nature a little while ago. I'll just give you one brief example of magistral production of drugs. Think of cytotoxic T cells to treat cancer. So some of you may be familiar with this. I'll just give you the brief outline of how this works. You identify a cancer patient. You maybe pluck out some cells from their blood if they have leukemia. Let's say you try to identify neoantigens in those patients' tumor cells, and then create cytotoxic T cells that'll go ahead and target those neoantigens. And you can modify the cells in vitro to be sensitive to these neoantigens. And the point is, everyone might have a different kind of neoantigen profile. So making a stockpile of cytotoxic T cells that target specific elements might not work if someone's tumor is due to mutations that no one else on the planet has. So the creation of cytotoxic T cells falls into this magistral production because you need to get the information from your patient and then right then and there figure out how to craft a treatment for that patient. So this magistral production of drugs kind of falls into that little workflow that I described. Okay, So DNA sequencing, how can that proceed? Well, again, we have experts on the phone like Radha and Kelly, and many of you have had lectures before, but I'll just give you my take on things and how this works in the context of this workflow. So if one has, say, an individual that has an idiopathic disease, and idiopathic just means hard to diagnose. And every hospital system on the planet has some number of patients who come into that system where their diagnosis just stumps the physicians. And here I'm not talking about really vague symptoms, more neurological or psychological symptoms. I'm talking about debilitating conditions. And here's just a few patients, I won't go through them all, that had kind of an idiopathic condition that was treated through the little workflow I described. I'm just going to mention Lily Grossman because she lives in the area, and as Kelly and others might know on the call, we spent some time sequencing Lily's genome, trying to find what's wrong and come up with various therapies. Sadly, although we had some success treating the rather debilitating condition Lily has, still there's not a complete success, and we're still working. But here's how this works. You have a patient and you sequence that patient's genome. I've drawn things as though we're looking at a haploid genome, but just bear with me for the sake of argument. And these bold letters here just correspond to variants in the individual patient's genome. We then compare that patient's genome to a reference panel of genomes. And if we make two assumptions, that the pathogenic variant that's causing our patient's disease is novel or unique to the patient, which is not an unreasonable assumption if we're assuming the patient has an idiopathic condition. So if they're the only patient on the planet Earth that has the condition, it's probably due to something that they have and nobody else has, right? We're also assuming that the variant, because of the severity of the symptoms, is highly penetrant. So if we make these two kind of assumptions, then we might be able to rule out as candidate pathogenic variants any variants that are seen in the reference genomes that we compare our patient's genome to. So in the little cartoon right here, this variant right here, sorry, this arrow is just a little off, looks like it could be a candidate. Another way we could argue that this is a candidate is by knowing what functional element in the genome that variant resides in. This could be a coding region of a particular gene, it could be a microRNA binding site, or it could be who knows what, and we'll go through that in a minute as well. So the basic idea is to kind of search for the needle in the haystack. You start out with all the variants, get rid of the ones that aren't novel, then ask questions about those that appear to be novel to the patient. Are they damaging of some functional element? Are they in a pathway of relevance to the condition and the symptoms? Are they expressed in a tissue of relevance to the condition? If you know a little bit about the genome and how to annotate variants, you can use various filters to maybe hone in on pathogenic variants using various techniques. And many people, including my group, over the years have developed tools. Ali Torkamani, who some of you may know, is at Scripps, developed a number of tools for facilitating this kind of identification of pathogenic variants. But these papers that we published are now dated. There's a whole cottage industry out there to identify functional variants. One very important point in this kind of workflow here is the composition of the reference genomes. And those of you who have had lectures in kind of population genetics will know what I'm talking about, but it's very important to make sure that your reference data set has the same, has people in it that have the same ancestral background as your patient. If your patient is, say, from the Aleutian Islands and your reference genomes are from people in some rural community in Africa, then there may be all sorts of variants that are shown to be unique to your patient and not in your reference genome that aren't causal. It's just you haven't compared your patient's genome to ancestrally matched reference panel genomes. So this is crucial. And fortunately, with the costs of sequencing going down, the size and diversity of reference panels is growing to facilitate this kind of discovery effort of variants that might be responsible for someone's condition. Okay, here is kind of a cartoon of a gene. It looks a little fuzzy on my slide. I apologize for that, but it was just taken from a textbook. So this just points out all the elements within and around genes that, if perturbed by, say, nucleotide substitutions of one sort or another, could damage or even enhance the function of those elements. And with computational tools alone, one can make fairly accurate predictions of the functional effects of nucleotide substitutions in all these functional elements. Now, this is not an exact science. You won't get it completely right, but it's better than flip of the coin. And you might say, well, why resort to computational tools? Well, again, as many of you know, any genome may have thousands, if not tens of thousands, of rare or even de novo or unique variants to the individual in question. So if you find that many variants in someone's genome and you want to know what their consequences are, subjecting each and every one to some kind of assay in the lab would be an expensive proposition. Although, as I'll say, we're getting better at doing this at high throughput. So computational techniques can at least prioritize variants in some way. Okay, and I won't go through this in detail because this paper is now a little bit dated. But for example, many programs try to predict the functional consequences of non-synonymous C-SNPs, coding variants, using, among other things, the conservation of the nucleotide across species. We published a paper where all we did was add a little wrinkle in this, and we used all the structural data out there to identify folds and loops and other structures in the proteins that, when perturbed by specific amino acids, would likely cause the dysfunction of that protein. And when we added this information, our predictor worked better than some of the others out there. Now, again, this is a little dated, and as I said before, there are a large number of groups out there that have made very sophisticated tools for kind of predicting the functional consequences of variations in the human genome to facilitate that filtering process, among other things that I described. You know, there's many tools out there. I won't go through these. EMBL has tools where you can predict the likely functional effect of variants, but there's many, many other tools for doing this. I mentioned Lily Grossman, who had this very severe condition. We used this filtering strategy that I talked about and ultimately subjected it to a number of little filters. There's Ollie and some other people that were involved. And at the end of the day, kind of came up with 5 candidate pathological variants, and ultimately settled on this ADCY5 variant as the most likely candidate, subjected it actually to lab assays. This is published, and came to the conclusion that this was, if not the causal variant, at least one that was contributing to her condition. Okay, now we were making the assumption that there was sort of a whopping mutation somewhere in the genome that was highly penetrant that was causing her disease. The vast majority of, say, tests out there, at least for cancer predisposition syndromes, but virtually all diagnosis instruments out there, focus on the presence of, you know, large effect variants in the genome because you can't. Sort of imagine looking under the light from a lamppost, you find these big effect variants. It turns out that of all the available tests, at least in the cancer context, to make a diagnosis of someone's predisposition to a rare cancer syndrome, collectively, they only capture less than 1% of all people out there. So even though they're useful, they only cater to incredibly rare disease. But what we're learning is that many diseases are not caused by single variants, as you know from your other lectures and your reading, but actually the cumulative effect of variants, each with small effects. And the recent paper by Kara showed that one could actually assess someone's risk for disease, if not diagnose their disease, by generating what are referred to as polygenic risk scores. And in the context of cancer, you could see that polygenic risk scores would capture about 1.5% of the population out there that are susceptible to breast cancer with a threefold or greater risk, right? And that one cancer collectively explains twice as much as the risk for one cancer as all the cancer predisposition syndrome monogenic variants do. So I don't want to pretend that everyone's disease is due to a monogenic variant that can be identified with the filtering strategy. But you got to start somewhere. So focusing on patients with identifiable mutations and pushing them through that little workflow I described makes sense. But I don't doubt in the future we'll be able to characterize people's polygenic backgrounds, understand what those each and every perturbation do, upset particular pathways, and on that basis, try to identify drugs. Now, one other thing I'll just mention is with respect to polygenic risk scores, we know that diseases are correlated. And I won't go through this in great detail, but just say that if someone is susceptible to cardiovascular disease, then the reason they're susceptible is because maybe they have genetic perturbations that upset various pathways. Those pathways, when upset, could also cause other diseases, leading to what people refer to as genetic correlations between diseases. This is incredibly important, not only for making a diagnosis, but predicting someone's trajectory with their disease, so their prognosis. If one is susceptible to a cancer based on having a collection of variants that also predispose them to heart disease, then the lifetime care of that individual might want to take that into account. Okay, now there are complicating factors in identifying pathogenic variants in someone's genome. I'll just talk about a couple. One has to do with the fact that humans are diploid, right? So oftentimes sequencing efforts, say standard 30x Illumina sequencing, is pursued by sequencing the pool of maternally and paternally derived DNA. And on that basis, you can identify sites in genomes that are heterozygous. So let's say in this example here, here's someone's stretch of sequence, and they're found to have a T allele and a C allele at this position, and a G allele and an A allele at this position. And let's say those positions are close to each other. If, say, the bold alleles actually cause disease, right? So these are disease causing. We won't know because we won't be able to separate the maternally and paternally derived sequence, right? Whether the two disease-causing alleles traveled together, say on the same haplotype, maybe from the maternally derived homologue of the gene, or whether they were actually dispersed, one of the disease-causing variants on the maternally derived version of the gene and the other variant on the paternally derived homologue of the gene. If it's the case that having, say, a wild type version of the gene can compensate for the perturbed version of the gene that has the two variants, then there won't be a disease. But if the variants have been transmitted, one on the maternally derived version of the gene and one on the paternally derived version of the gene, leading to the disruption of the function of both homologous copies of the gene, then you could have disease. And this situation is known as compound heterozygosity. And many rare diseases are attributable to compound heterozygosity. We had a review about this a while ago, but again, there's many papers that describe this. And the point of this is, unless you can resolve what is referred to as the phase of the genome, the maternally derived nucleotide content versus the paternally derived nucleotide content of the sequence, you won't know which of these two situations occurs. And again, if it's the case that you need compound heterozygosity to get the disease, you would need to resolve phase in order to prove that there's evidence for compound heterozygosity. So just a complicating factor. As I mentioned before, a couple of recently published papers have kind of gone through the genome, just counting up the number of instances of compound heterozygosity that exist. Another paper, I think this has since been published by Dan MacArthur's group up at Harvard. But what they did, again, was just catalog all the instances of compound heterozygosity in someone's genome, and it was substantial. And they also found that some genes had a preference for having variants, two variants, one on maternally derived versions of genes and one on paternally derived versions of the genes. The explanation for that was a little bit vague, but suffice it to say they found that some genes had a greater frequency or seemed to be more amenable to this compound heterozygosity phenomena than others. Now, there are many other genomic factors that contribute to disease besides the inheritance of, say, single nucleotide variants, even in the compound heterozygosity setting. For example, structural variants can cause a disease. De novo variants can cause a disease. So variants that no one else on the planet has because they arose in the meiosis or transmission of the variants from a mate pair to an offspring, right? There are many, many other genetic contributors to a disease that one might have to tease out that can be complicated. I'll mention mosaicism as one. So there are often rare diseases where some fraction of cells in the individual harbor a mutation and the other ones don't. You know, maybe this occurred through some post-zygotic event or whatever it is, but the reason I'm pointing this out is many of these phenomena would be hard to dig out with kind of standard 30x sequencing. You'd need to subject the sequencing to other sorts of things, right? Like phasing the genome for compound heterozygosity. To really know if something is de novo, you might have to sequence parents. So I'm no way saying that this identification of variants that might cause someone's disease is easy, actually pointing out the challenges, but people have had success. Okay, another interesting phenomenon that people should be aware of in trying to understand what the genomic underpinnings of a disease are have to do with cancer. And this classical two-hit model that is enjoying a little bit of resurgence among the biomedical science community. And I'll just point out how this works. So let's say there's a mom and a dad here and they have a child. Dad had a mutation on in one copy of a particular gene that he transmitted to his child. Now, because the person is heterozygous, the wild type copy of the gene can make up for the perturbed copy of the gene, so the child is otherwise healthy. But then somatic cell replication occurs, and during somatic cell replication, let's say that a mutation arises on what was the wild type version of the gene in a subset of cells that ultimately has some kind of selective advantage. What this could lead to is cancer. And there are some forms of cancer where this really strange form of compound heterozygosity, where you have a combination of an inherited mutation with a somatically kind of acquired during cell replication mutation occurs, such as Lynch syndrome is due to this unique combination of variants. Now here, what you might need to do is not just sequence the germline, say in a cancer patient, but also the tumor DNA and go ahead and look for evidence for compound heterozygosity, where you have this somatically acquired mutation coupled with an inherited mutation in two homologous copies of a gene. So again, another complication kind of throws a monkey wrench in this workflow in understanding what is causing a patient's disease and how to treat it. Okay, so once you identify a variant, what can you do? Well, this is not trivial, but just suffice it to say that if one could identify variants that are causing someone's disease, understand that the development of drugs to treat that disease might be facilitated by that genetic understanding. This makes a lot of sense for obvious reasons. If you know what's causing a disease, you know how to target it. But in the drug development community, it's been shown that drugs that target genes that ultimately are known to harbor variants that cause the disease that those drugs are trying to treat or prevent ultimately do better than drugs that are designed to target genes to treat a disease where the targeted genes don't harbor variants that cause disease. So again, this makes a lot of sense. This paper kind of shows that genetics can really help identify more solid drug targets. Now, again, you're no stranger to all sorts of resources on the web where one can just look for genetic variants that are known to cause disease and gain some insight into what functional elements they might reside in and what they do. The GWAS catalog, OMIM, there's many databases for this. I won't bore you with all the details. Now, another way of going about finding whether or not you have a mutation that's causative of a disease is to make constructs. Kelly Frazier's an expert on this. You can make induced pluripotent stem cells that harbor variants, right, causing disease. Here's a collaboration I have with Stuart Lipton right across the street at Scripps, where he made organoids from iPSCs from individuals that had rare PS1 mutations. PS1 mutations are known to cause a form of Alzheimer's disease. And then he subjected those organoids to different kind of molecular phenotyping, electrophysiology being one, and showed that when he contrasted the mutant-bearing organoids of cellular phenotypes, electrophysiologic phenotypes, to cells where he used CRISPR, kind of genetic engineering techniques, to change that mutation into what is a wild-type nucleotide, he showed phenotypic differences. So we were able to understand what this mutation was doing at the molecular level, right, using organoids and other things. So again, many people at UCSD, again, Kelly's lab, Larry Goldstein's lab, Alison Motri's lab, many labs do this. So you should be no strangers, but you could see how the use of organoids could fit into this kind of workflow that I'm describing. Have the genomics guys find mutations, hand them off to the labs that can make organoids and look at the molecular consequences and go from there. Okay, I won't go through this. Ultimately, what Stuart was able to identify based on his electrophysiologic characterization of the phenotypes in the cells is that there was a lot of calcium signaling disruption going on in the mutant-bearing cells. The kind of highlighted regions of these cells just indicate calcium signaling. So that meant that if he could identify a drug to kind of control the unchecked kind of calcium signaling, he might be able to treat the disease after understanding what the causative mutation is and what it's doing. Now, doing assays to understand what the functional consequences of mutations are at scale is being pursued. Former colleague of Kelly's and mine, Ryan Toohey, did this with a group at Harvard, developed an assay where they could, essentially evaluate the functional consequences in terms of the expression levels of the genes for massive numbers of naturally occurring variations, right? And there's other tools out there that could be used. Interestingly, there's a new database called MAVE database where a group of researchers, including Jay Shanduri's lab at the University of Washington and others, where what they do is they take a particular gene and surrounding kind of cis-acting functional elements and systematically perturb each nucleotide in that sequence, and then subject cells that have those perturbed sequences in them to a wide variety of functional assays. And then they can have kind of a big table that says what each variant might have induced in terms of molecular physiologic consequences, and they have a database. Now, these databases are going to grow in numbers, as you can imagine. So if one identifies a mutation, say from sequencing, and you're wondering if it's likely to be a pathogenic variant, you might be able to go and look up what the functional consequences of that nucleotide substitution or deletion or whatever it is in databases like this, or databases that house information from these large-scale studies. Okay, so moving up the kind of workflow here, if you've identified, and I'm not, again, in any way saying this step is easy, right? It's just tools are being developed to facilitate it, allowing one to go from kind of start to finish, or at least make an attempt to go from start to finish. But if you've identified, say, a variant that induces a molecular physiologic perturbation, and you now have insight into what is creating or contributing to the pathology, how do you go about identifying a drug that might combat that pathology? This, again, is not trivial. As I mentioned in the context of the work we're doing with Stuart, and there's many other labs doing similar work, we knew that calcium signaling was kind of the issue. So he built a few constructs to see how they might kind of ablate or deal with these calcium signaling perturbations, and identified one drug, nitrosinapsin, which is actually a combination of two drugs. Won't go into the details, but it looks like it might actually correct the defect in some way. And since this is a combination of two available drugs, it might be possible to test the drug out in repurposing efforts. If both of the drugs that were combined to create this combo drug are approved for use, then it's easier to get their combination approved and ultimately tested, say, in patients that have PS1 mutations. Okay, but there's a growing number of resources that one can tap into, such that if one had, again, a variant that was known to induce a particular perturbation that might explain someone's disease, you could go see if there are any drugs out there, or compounds, not even approved drugs, just compounds that, say, haven't been taken to the next stage in development, that might show promise for overcoming those perturbations. I won't go through these in great detail, just say they're growing in number. There's this paper right here, open source drug discovery platform that enables ultra large virtual screens, was in Nature just a couple of editions ago. It's a complex paper, but it's open source. So if you learn how to use the interface, you can query that database for drugs on the basis of some target that you come up with. The links effort, costing the taxpayer, many millions of dollars, is to build big databases that one can search in similar ways like this. And there's other kinds of strategies out there. I've collaborated with people to make tools for this. One I want to share with you revolves around what is known as the connectivity map. So if one has, say, a gene expression signature, so in the context of having a mutation, maybe you've introduced the mutation into organoids, like Stuart Lipton had done. And your molecular characterization of the effect of that mutation is just looking at gene expression patterns, say expression patterns that differ between the wild type cell and the mutant bearing cells, right? And if you find a particular signature, gene 1 is overexpressed by 60 units, gene 2 is underexpressed by 2 units, gene 3 is underexpressed by 13 units, et cetera, et cetera. You have kind of a signature associated with the mutation and its effects, okay? Then what one can do using the connectivity map and other sources, this is in links and other things that I had mentioned earlier, you can then query these databases to see if there are drugs out there that essentially reverse this fingerprint or signature induced by the pathogenic variant. Now, why would you want a drug that reverses that signature? Because if this is the expression pattern that's known to accompany the mutation, and that mutation is causing pathology, then these expression changes are the things that are reflecting the pathology induced by the mutation. So if you could find a drug that essentially reverses the signature induced by the mutation, that would be a candidate drug that you could test out. And the connectivity map and the related resources are basically made up of cell lines that have had their expression levels measured at baseline, and then after they have been stimulated with a wide variety of drugs. I don't know the total number of drugs that the LINCS database has in it, but let's say it's on the order of thousands, if not tens of thousands, right? And you have different cell lines in there. So you could then look for consistency in the effect of drugs across the cell lines in reversing a particular target signature. Okay, and so in this little cartoon example, we see that compound 3 is known to decrease the expression of gene 1 by 51 units, increase the expression of gene 2 by 4 units, et cetera, et cetera. And if you just look to see how those changes might counteract the changes associated with the mutation-induced fingerprint, you can see it looks pretty good. So there's your candidate compound. That is the intuition behind the connectivity map. And it could facilitate the discovery of drugs, repurposing or novel compounds that might need to be developed further for combating a disease. Okay, and I won't go through this, but you might find that there isn't one drug that reverses the signature entirely, but maybe two drugs in combination appear to reverse the signature. Okay, so here are the resources here that you could look up if you're not familiar with them. There's other sources to look for drugs that kind of match genomic perturbations. I won't go through that. A number of other DASes, the therapeutic target database. You could put in, EGFR if you know you're treating a cancer patient, say with an EGFR problem, what drugs are out there that treat or let's say target EGFR. Now, you'd have to know whether you want a drug that is an agonist or an inhibitor, but you could hopefully identify drugs that induce effects in the direction you'd like them to go in. There's a number of other resources, so if you wanted to do this more signature searching, tons of resources exist for doing this as well. So again, I'm pointing out that this little workflow going from start to finish, one could gather together the different tools that are out there for each and every stage and kind of put things together to hopefully arrive at a solution to the patient's problem. There's other tools out there. I won't go through this in detail, but just point out that now digital therapeutics are approved for use. And what digital therapeutics are just iPhone apps, let's say, that are designed to deliver content to deal with someone's problem. So smoking cessation is one where digital therapeutics have been approved for use. So essentially, if you want to stop smoking, this is a little bit off topic, but just because it's interesting, if you want to stop smoking, you go to a psychiatrist, they could prescribe you an anti-anxiety pill, get you to take Chantix, wear the nicotine patch, do all sorts of other things that have been shown to be somewhat efficacious in treating smoking addiction. Or what they can do now is literally prescribe you an app. And that app would just coach you through your cravings for a cigarette using different tricks, gamification, imagery. So if you have a craving and you press the little button, maybe you're immediately sent a picture of a burnt out set of lungs. And that is going to cause you to say, I don't want to smoke because that's what's going to do my lungs. Or you'll be sent a picture of a brook flowing gently through a forest to kind of calm you down. So there's all sorts of ways that one might be able to deliver content to deal with certain issues. These are becoming more and more popular for mood and mental disorders. Another reason I brought this up is you may have seen that the FDA approved accelerated use for digital therapeutics to treat mood and mental disorders that are associated with COVID-19. So they kind of did fast track on the approval of the devices because these are the perfect social distancing. You don't have to go to your pharmacist. You can download your app to deal with the stresses and strains you might be suffering from. Okay, so moving on up this workflow. So we've maybe identified a drug based on everything more upstream in the workflow. Now we have to test it. And here, if you only have one patient, how are you going to see if the drug works? Well, this is in the area of N of 1 trials. I've had the opportunity to interact with the FDA and other groups about N of 1 trials and their utility. I'm not alone in this. I'm just showing you this because I was involved in some of these discussions. But here's how an N of 1 trial essentially works. Let's say we have some phenotype, blood pressure, that we want to control. And we don't know the best way of doing that, but we have a candidate drug. What we could do is measure someone's blood pressure at baseline, give them the drug, take them off the drug, give them a comparator drug, and do that, collecting blood pressures continuously, potentially using devices and other sorts of things. And on the basis of what we actually observe or the statistical analyses we could pursue on the data, we might come to the conclusion that, say, drug 1 is better at lowering this patient's blood pressure than drug 2. We've only done this on one person. It could be that other people have a different pattern of response. And it could be that there are other phenotypes we could have measured that accompany blood pressure, heart rate, BMI, sleep quality, you name it, and see if the drug affects those as well. N of 1 trials like this have been done by many groups, reviewed in a couple papers. We've described how one can design these studies in sophisticated ways. Just for the interest of time, I won't go through this. I had mentioned that kind of wireless devices are useful for collecting relevant data in pursuing these sorts of studies, right? So again, you may have a blood pressure monitor or something you could actually slap on over someone's heart, a Band-Aid-like device that just sits on someone's heart and is continuously measuring blood pressure and heart rate while you're giving them different drugs, say, 1 week, some antihypertensive, another one, a different one, and then sort of in a way that is more or less transparent to the patient, making determinations on whether your candidate drug is working. Now, this won't work in all instances. If you have a severe life-threatening condition, you won't have the luxury of time to kind of play with all these devices and test out different drugs. But suffice it to say, in some instances, this can be done. I go back to Lily Grossman. The family actually collected data on some phenotypes that were characteristic of her pathology, like she had tremors at night. And this is a year's worth of tremor readings. You can see she had a number of tremors. We used a little device. We gave her an actigraph that she could wear when she was sleeping to see how severe the tremors were, so they wouldn't have to be recorded manually. And you see we had good correlation. What we did, or what I should say Ali Torkamani did, which was published, is take information about this ADCY5 variant that I argued we identified as one pathogenic variant. He then went and looked through various databases of the type that I described and found a candidate drug that we thought could benefit her nighttime tremors, and did this little N of 1 study. And if here was the number of tremors and severity of tremors before we gave the drug, which we thought would match the pathology induced by ADCY5 variant, and here is the number and severity of tremors after the drug was given. I mean, we didn't even need to really do any statistics on this or anything fancy. It just looked like the drug was doing the trick. Since then, the family has been advocating for treatments like this for patients with similar conditions. We've done other N of 1 studies. I'll whiz through this. Here's one where we had a patient with a sleep phase disorder induced by a particular genomic clock gene variant. They couldn't sleep during night. They're up all day. So we measured their sleep quality while they were on the medications that they had been prescribed, so on multiple medications. This really shows the danger of polypharmacy. But because the patient was not happy with their sleep phase disorder, they were being treated for depression, insomnia, other conditions. So what we did was we measured their sleep quality while they were on different combinations of the drugs that they were prescribed. And what we ultimately found, which was a little ironic, is their highest quality sleep was they were on fewer drugs. So we actually learned the opposite. Many of the drugs that they were on were actually probably compromising quality sleep and not improving it. And this was published. Another one we looked at where we had a patient with a hypertensive disease. We gave them a couple of different drugs, measured blood pressure continuously to the degree we could in this patient. It looked as though drug B here was lowering blood pressure a little bit more than drug A. So we were about to cry victory and say we found the optimal drug for this patient. Unfortunately, we looked at some important covariates and found out that this patient lost about 7 to 10 pounds during this period. So we couldn't attribute the blood pressure lowering to the drug, but to the weight loss. So what ultimately turned out to be an instance where by participating in the study, the patient actually grew more health conscious and in that way controlled their blood pressure. So there was no need to treat them with the drug. N of 1 studies like this have been shown to have clinical utility, even though they might cost money to pursue. I won't go through this, just to say that in some studies comparing how N of 1 trials fared with, say, just the guesswork that goes on in people deciding what drugs to use, in at least a couple of instances, there was actually cost savings. So even though there were upfront costs in pursuing the N of 1 trial to convince physicians that the drugs they thought would work did work, those costs actually were made up for by a lack of return of the patient to the health system complaining about their drug not working. So there was actual savings of costs to the health system, even though there were initial upfront costs in conducting the trial. Now, another way of going about trying to identify therapies is simply to match drugs to particular perturbations. So in cancer, as many of you know, there are multiple aberrations that are found in patients' tumors, and there are drugs that have been specifically designed to counteract those problems. So the most famous one is if some people have a form of leukemia caused by the BCR-ABL gene fusion, then Gleevec was designed to kind of overcome that defect. So if you're diagnosed with this particular form of leukemia, you want to be given Gleevec. Now, if you have a different form of leukemia that's not caused by this fusion event, then Gleevec might not be the optimal drug for you. If you have breast cancer that shows HER2 overexpression or amplification, then Herceptin, because it's an inhibitor of the HER2 gene, should be given. So the community is identifying kind of matches to different perturbations. Now the problem is, no one is going to conduct N of 1 trials for every patient to do this. So this poses a dilemma because cancer is a more common condition than, let's say, the condition Lily Grossman had. So how are we going to deal with matching drugs to patients in this context? Well, the idea is if we have patients and we can profile at them some level, sequence their tumors, and we have a bunch of drugs, we need to come up with some kind of algorithm to match perturbations in the tumor with the drugs, and then treat the patients with the appropriate, in this instance, color-coded drug, okay? So problem is conducting the trials to show that the matching works. So I should emphasize that what is actually going to be tested in trials are not the effects of individual drugs, nor the profiling assays. It's how the matching is done. Let's say that two groups are given data, profiling assays, links data, cell perturbations, organoids, other sorts of things with mutations identified in them or genomic perturbations of another sort, and then they have activities of drugs from connectivity map. It could be that two groups come up with different matching algorithms. One says that the blue-colored patient should actually get the green drug for whatever reason. They're based on their own biological insights or analysis of data, as opposed to the blue patients getting the blue drugs. So the matching algorithms here are ultimately what's at stake and what are being tested. And many trials are being pursued to test these kind of matching algorithms, and they go by various names: basket trials, umbrella trials, adaptive trials. So in the context of our workflow, let's say we don't just have one patient like Lily Grossman, we have multiple patients where we've identified different perturbations they have, and we want to simultaneously test our ability to match drugs to each one of those patients based on their genomic perturbations. Then what we would be doing is ultimately testing a scheme for matching the drugs to the patient's profiles. And there's, again, a lot in the literature describing how this could be done. Many of these trials are being pursued in cancer. I'll just mention one, the Shiva trial, because it's interesting in that it didn't work. And so it set the field back a little while. So the Shiva trial had 10 different treatment buckets. So they matched profiles from patients to 10 drugs that they had and then treated them with the drugs that each patient was allocated to, the little bucket. And then they compared the ability of the matching scheme, right, to result in better outcomes than whatever the standard of care was. And what they found is that there was no real difference between the two groups. And as I said before, a lot of editorials were written. Here's one on how precision oncology, discovering something about a patient and matching a drug to it, wouldn't work because it's just too complicated. And they used the lack of result from the Shiva trial as evidence for this. Now, interestingly, this raised questions. It could be that the 10 drugs they used do have utility. It's just the matching algorithm they used wasn't good, right? So again, we could think of another group that says, you know, these 10 drugs, they should be given to the patients, but not in the way that the Shiva trial had assumed, but in some other way. So maybe the trial failed not because precision medicine is an illusion or that the drugs weren't good, but that the matching algorithm didn't work. And in fact, in follow-up papers on the Shiva trial, they did some ad hoc, kind of post hoc analyses of the data and found that had they matched drugs in a slightly different way, they may have seen different outcomes. Now, algorithms are being described for matching drugs to patients all the time. These are now dated, but they kind of tell a story. Patrick Soon-Shiong, the world's richest physician who now owns the LA Times, as many of you know, has companies, Nant Health being one, where they have claimed they've identified ways of matching drugs to cancer patients' tumors that could lead us to the nirvana of coordinated care delivery. These are his quotes, not mine. And, you know, not to be outdone, around the same time, guys on the East Coast, because Soon-Shiong's in California, had said maybe we'll start using IBM Watson to identify drugs that match patients' tumors. Now, what would be very interesting is to know whether the rules or matching scheme used by Nant Health was similar to that being exploited by Watson. Because if they weren't, then you could run into that situation I described in the Shiva trial where the algorithm for matching the drugs was a little bit off, right? So a good question to FDA officials is whether we will have to have kind of large-scale clinical trials comparing the efficacy of different intervention matching schemes or algorithms like this. And I've had the opportunity to talk to the FDA about this. You can imagine how complicated this would be. Now, there are many people who say, you know what we should do? When we start to push drugs through the different phases of development, once we find out that a drug is kind of efficacious, let's not do these really large-scale clinical trials. Let's just put them out on the market. And then let the use of those drugs and profiling of patients decide who benefits from them. So essentially do away with the expensive billion-dollar phase 3 and phase 4 trials. Now, I wasn't the advocate of this. Two people, Amy Abernathy and Sean Cozen were, and they described this in the Wall Street Journal and some other peer-reviewed publications. And what I want to point out is Sean Cozen, at the time he wrote this, was at the FDA. Amy Abernathy wasn't. Now Sean is at a pharmaceutical company and Amy Abernathy is at the FDA. So these are two regulatory officials who are actually advocating for maybe replacing phase 3 and phase 4 trials with something else. And recognizing that these large phase 3 and phase 4 population-based trials, if they're not profiling the patients to identify which subpopulations might do better on those, are actually flawed and shouldn't be pursued. So we should just get the drugs out there, profile patients, and see which ones work. In order to do that, you need to develop appropriate infrastructure. Some infrastructure is called a rapid learning system. I'll walk through this briefly because of time constraints. You have a bunch of patients, you collect data on them. Maybe these are tumor profiles, maybe these are germline profiles to see if they have ADCY5 mutations or not. You then build predictive models based on this data, like who is likely to respond based on all the perturbations, and then you test it out on data. And it might be the case that it doesn't work perfectly. You get some false positives and false negatives. Now, most studies would stop here, publish the results, and say, well, we'll let someone else figure out how to deal with the false positive and false negatives and improve the predictive modeling. But with rapid learning systems, what you do is you collect more patients, knowing that your model wasn't perfect, add them to the data that you had before, refit your model, and then test the predictions with new data, and do this in perpetuity. So essentially what you're doing is continually evolving a system that should identify subpopulations based on their outcomes, right, that match drugs to patient profiles optimally. And there's lots of people who have discussed this in the literature. And many people have pointed out that such studies could be facilitated using profiling of many sorts with AI techniques. I won't go through this, just say, you know, many people have found out that, say, their machine learning based predictions of who will and won't respond to a particular drug do better than a panel of experts. Okay, so in conclusions, personalized medicines will require new vetting and testing strategies. Emerging technologies are changing the landscape of healthcare. Understanding the factors that should determine or mitigate treatment effects for individuals will require very integrated approaches. I said things could be framed in the context of this workflow from going from start to finish in patients. But if you start to scale that, you might run into some of these issues about testing intervention matching schemes. Focused patient-oriented studies and aggregated N-of-1 trials will be more revealing than traditional clinical trials. I'm not alone in this belief. I think I showed you some evidence that even the FDA is sympathetic to this. Matching drugs to individual patients will be key to the future, but vetting those matching strategies will not be trivial, and maybe we'll have to resort to learning systems where we aggregate electronic medical record data from multiple health systems, building up a monster database that could be mined for patterns, dictating or showing who may or may not benefit from particular drugs. And I'll just stop there and point out some funding sources. Two of my students just graduated this month, and then have some other people. So thanks, and I can, I guess, stop sharing, or Kelly, Radha, let me know. What do you guys want to do?
Well, I think a good point would be to see if any of the students have any questions about this. It was a tour de force. Thank you, Nick. It was great. So do we have any questions?
Yeah, I have a question. Thank you for that talk. That was really interesting.
And who is speaking? You didn't pop up on my screen.
Oh, it's Stephanie Heimler.
Hi, Stephanie.
Is my video not working?
It might be. You can see her? Okay, good. That's all that matters. Go ahead.
Oh, well, as long as you can hear me. Yeah, I was interested in what you said about the molecularly guided therapies a few slides ago and why that trial didn't work with the matching. And I was wondering if you knew, with all those different patients and the different drugs from the different baskets they tested, were those all new investigational drugs that were maybe perhaps used with other therapies like chemotherapy or other drugs that in combination made them fail, or were these all used alone as, I guess, as new drugs to be investigated for their individual tumors?
Those are great questions, and I would encourage you to kind of read this. I think one of the papers that's going to be described is going to be talking about trials like this and maybe some of the issues, so we may know more or learn more on Thursday. But in this trial, yes, the patients were treated with one or another of the 10 drugs. And if they had been on therapies prior to their inclusion in the trial, that would have been an important covariate to take into account, right? How well were they doing? My guess is if they're enrolled in the trial, they wouldn't have been doing all that well, because no physician is going to take someone off a drug that works just for the sake of an academic exercise. But your point about whether or not combinations could be used. So you could imagine an intervention matching scheme of the type that was tested here, where the buckets don't include a single agent, but combinations of agents, right? And that would make the trial even more complicated, because instead of 10 treatment buckets, you might have hundreds of treatment buckets, which is exactly where we're going if we're going to try to personalize medicine, right? We're going to identify smaller and smaller subgroups. So, I guess sadly for me, but your generation is the one that's going to have to deal with these problems. And they, I think, are going to become more pronounced. But given the availability of high throughput computing, databases like the ones that are being built, you'll probably have an easier time than, say, we are currently. But those questions are a good one. Another thing that maybe I should have mentioned was that, they assumed when they enrolled their 100 patients who would be matched to drugs, they'd roughly have 10 patients per bucket, right? And they did this to purposely kind of give equal opportunity for each drug to show its trick. But it turns out, if you look through the data, some buckets were filled with more patients than others. Now, let's say that of the 10 treatments, 9 of the drugs work well, but 1 drug doesn't. It just doesn't do the trick for the people that they believed had the appropriate profile to get it. That could water down the effect of the entire matching scheme, right? Because if the one drug is not working and more people are assigned to that bucket just by chance alone, maybe 15 out of the 100 people instead of the expected 10 were put into that bucket, and that bucket's drug didn't work, it compromises the evaluation of the whole scheme. So these are statistical issues, they are design issues, they are profiling issues that are raised by these trials that the contemporary biomedical science community is dealing with. But they are, your questions are spot on. And I would encourage you to look through this, because this, is the future. This is all about precision medicine. So this kind of approach is not going to go away.
So with the model, I guess in light of the model you were just describing with the statistical rapid learning, do you think it would have been appropriate in this case if some of the patients who already took drugs in the baskets where you said, it may not have been helpful for them, could those drugs be reallocated to other patient groups, or does the model just account for, I guess, starting over with new indicators?
Yep. That's a great question too. So, when you build these predictive models, right? So you have a bunch of patients, you want to know how to treat them. And so you're going to build a model that is going to be predictive of who should get what drug. What you include in this model. I mean, I made this model as simple as possible, but you could imagine having, hundreds of covariates here, predictors, one of which might be prior therapy. One might be, other diseases that the patient suffers from. One thing that I didn't go through was imagine you have a cancer patient, right? And you want to profile their tumor and come up with an insight that suggests you should increase the level of the ACE gene or something like that. Based on the tumor biology, it looks like you'd want to stimulate ACE production. Let's say that's the case. We know that ACE overexpression could also complicate someone's blood pressure level and increase it. So if the patient just also happened to be suffering from hypertension or was susceptible to hypertension, then even though you have a molecularly guided therapy to treat their tumor, you don't want to kill them by giving them a drug that's going to give them hypertension. So clearly there are complications here that need to be worked through, but at least the intuition is that with enough data, the number of predictors you'd have there, one might be, does the patient have hypertension? And then you have an interaction term between hypertension and treatment with the ACE drug, right? You could work this out. Now that is all in theory. And as I said, these models have been proposed. Some are actually being implemented. So the cancer link right here is something that ASCO, a big organization, is actually pursuing as we speak. So, they cater to the needs of many oncology patients, and they're collecting data on those patients to build systems like this. The degree of success they're having is, in the eye of the beholder. You'd have to read the papers, but I'd say we're learning things, certainly, but we're far from perfect.
Thank you.
So are there any other questions that people have?
For the end of one trials, there was a mention on the slide about patient well-being and not necessarily, I guess, like the more traditional outcomes.
Right. Yep.
In that case, is it possible to control for mild placebo effects?
Yep. Absolutely. And just going to this design slide, wherever it is. So when you design these trials, and again, some designs will really only work in certain situations. As I said before, if you have someone who has sepsis and is got 2 days to live unless you figure out a treatment, you're not going to be able to do what I'm about to describe. But if you have someone with a chronic condition like hypertension, where you won't compromise their health too much by switching drugs, right? Then you might be able to do something to address your kind of placebo effect, which is to include washout periods in your thing, or blind the patient to what drugs they're on, right? So let's say drug 2 here was a placebo, and you design the trial such that your patient was blind to what drug they were getting. So they wouldn't know if they were actually getting a placebo or getting an active compound. Then if you looked at their blood pressure and you saw their blood pressure drop as much while they're on placebo as they were on active drug, then you'd argue that that person there's a placebo effect going on. So placebo effects can be dealt with in N of 1 trials, but not in all settings, as I described. But your other question about patient benefit. So there is, as you probably know, if you take ethics courses or exposed to this, a real kind of hostility towards treating patients as kind of guinea pigs, and just saying, well, look, we want you to potentially put yourself in harm's way so we can learn how to treat future populations. There is growing number of antibodies to that kind of approach. So instead, what the biomedical science community is trying to do is pursue studies where the patient is a participant in the science, right? So the data from the trial is made available to them to the degree that they want that data or understand it. They are, like an active member of the scientific team. In that way, we could all argue that the research is being pursued not to settle on some academic question, or benefit future patients, which was clearly the case of clinical trials in the past, where people were more or less asked to volunteer them so that they could benefit the next generation. Now there's a sensitivity to pursuing these studies not only to gain some scientific insight, but actually benefit the patient, all right? And if you find out that the drug early in the trial is actually not benefiting the patient, then you stop that trial for futility and move on. So future trials are undoubtedly going to be pursued in ways that are more sensitive to the patient's needs as a participant and not just as a guinea pig.
That's great. Thank you.
Are there any more questions? So Nick, I have one about using the AI.
Yep.
So, there's lots of studies, and have you put forth, that's indicating that the AI is actually better, even in, a pathology situation,
right,
than experts. And so how do you feel? I mean, I know that you work closely also with clinicians. How do you feel? How well why isn't AI moving faster?
Yeah, it's a great question. And there's two kind of answers to that. And some are interesting and others are probably a little bit more academic. But my belief is that if you have predictive models based on AI, you present that material to a physician to support their decision-making, not replace their decision-making. I mean, that one kind of juxtaposition of words actually, calms people down. Because
Yes.
I think
Yeah.
no one wants to put blind faith in these more black box approaches to making predictions. However, physicians, as you know, from say participation in tumor boards, right? So if it's unclear how to treat a patient with cancer, it is routine to convene a tumor board where you have multiple physicians just review the case, check each other's recommendations to make sure they're sound, and on that basis come up with a consensus way of treating the patient. Actually, tumor boards are even used in the context of clinical trials, which creates problems because maybe the drug that was recommended by a matching scheme is one that the tumor board overrides for some reason. If you have that kind of discrepancy between what the matching scheme predicts and what the physicians ultimately settle on, you have to take that into account when you evaluate the utility of the matching scheme, right? Because so providing material to support physicians' decisions is the way I think we should go. And physicians draw on all sorts of things. If a physician is stumped on how to treat a patient, what are they going to do? They're going to go back to their textbooks and see what do I know about this weird neurological disease. They're going to consult their colleagues. They're going to call them up and say, hey, I'm not expert on this. You are, let me know. They're going to go to the literature. They're going to draw on all sorts of things to make decisions that ultimately could be considered algorithm-like. You know, who's to say that when you go to PubMed and you put in ADCY5 mutation, and you only have time to read the top 10 papers or abstracts of the papers, that the ranking system used by PubMed is the best for identifying the papers that a physician should pay attention to in coming up with the treatment decision. I mean, there's an algorithm there. It might be subtle, but it's there. So what you do is just argue that this is in supportive decision. The second part of your question is interesting in that many of these techniques, and we probably have some people in computer science in the class or have computer science experience, like deep learning. Deep learning is very much a black box. You have a bunch of inputs and then you have outputs, and you really don't understand the logic behind matching the inputs from the outputs. You do have people like at UCSD and Trey Eidecker who want to kind of use what they call visible machine learning, or where you can, understand what the little layers in your deep neural net are actually capturing, right? But suffice it to say that many of these techniques are black box, and no one wants to put their complete trust in something that they don't understand. In fact, as an
that
anecdote,
makes sense.
Yeah, as an anecdote, you probably know that the European Union banned products from being sold that were based on kind of black box models. So if you have a product that is taking some inputs and predicting something that is purely black box based, that you can't explain, right? You don't know how the weighting of the coefficients and all this stuff plays, then you can't sell that product. And the reason why they did that is they had this kind of nightmare vision where, driverless cars are now out on the market, and someone's in a car and it smashes into a tree and kills the person. And then they go to Elon Musk or whatever and say, why did that happen? And he goes, I don't know. We can't figure it out. It's the deep learning. That wouldn't satisfy anyone. So there is a push for kind of trying to understand the input-output relationships to the point where these sorts of confusions don't arise.
Perfect. Okay, great. Well, I know some people have to run to their next class, so let's give Nick a round of applause, either with our silent clapping hands or our real clapping hands.
Well,
and we appreciate it, Nick. And for the students, Rhoda and I would like to emphasize that the class evaluation is available, both the overall class evaluation as well as the evaluations for each of the individual instructors. It's really important that we get your feedback on multiple fronts, as we've discussed before. So please take the time to fill those out. Okay? All right. Thank you very much. And we will see you all on Thursday at 8:30, right?
Got it.
Looking,
Okay,
looking,
thanks.
looking
Thank you,
forward
Nick.
to it.
Yep, thanks,
Thanks.
Rhonda. Thanks, Kelly.
Thank you.
Bye.
Bye-bye.
Bye. Bye.